the membrane's folding inward, and whatever is being, might be bound to the membrane, it might just be pulled in as liquid-like. So it's molecular in size. Viruses are not much more than molecular complexes, so they can be taken in this way as well. And it turns out, again, it's not phagocytosis because viruses are non-cellular. They're much too small for that. But it turns out animal cells in general tend to take in materials from their environment. They're constantly sampling, taking materials in, and it is a way that they obtain nutrients, but they can also pick up virions. So because the viruses are taken in through endocytosis, when they get inside, they're surrounded by a membrane, they're inside a vacuole or a vesicle, and they come in as a complete virion. So their mechanism of entering is very different from bacteriophage. Typically, bacteriophage leave their packaging outside. They come in as just nucleic acid. But animal viruses don't. They typically come in as complete virions. There is a variation on this theme, and because there's one, there's probably more than one. But the human immunodeficiency virus, HIV, gets into its host by a mechanism that is a modified version. It's not typical endocytosis. There was a period of time where this process was referred to as viropexis, and then for whatever reason that term was discarded. It's no longer applicable. But what happens is these viruses have an envelope. As you recall, the envelope that's on some viruses is essentially cell membrane. They picked that up from the cell that they were exiting. So HIV is an example of an envelope virus, and what it does is bind to the surface of the cell membrane and then get in that way. So what you see here, an illustrated version, you see the viral envelope here. The viral envelope has receptors that interact with receptors on the cell membrane surface. There's the receptors that are on the virus are glycoproteins. They're referred to as GP120 and GP31. But this is the CD4 receptor, which is a characteristic of a particular type of cell that HIV can infect. We'll talk about this more when we get into viral diseases, but and more specifically about this particular type of virus. But the CD4 receptor is associated with a certain group of white blood cells that are called helper T lymphocytes. The helper Ts have this CD4 receptor, and it makes them susceptible to infection with HIV. The CD4 binds to the glycoprotein 120, and then there's another little protein over here called the CCR5, that's the chemokine receptor. That's also going to make contact with the virus. So here we see the CD4 receptor kind of bent over, and it's attached to the glycoprotein 120. And then here's the CCR5 also binding to that. And then the glycoprotein 41 kind of goes into the membrane. What it's going to do is cause this cell membrane to fuse with the envelope of the virus. So this is a very different mechanism for getting in. The membrane of the cell doesn't fold inward. It just binds with or fuses with the envelope of the HIV virus. So again, the viral glycoproteins are GP120 and 41. They're binding to the CD4 receptor and the CCR5. These are kind of details that were determined about how HIV gets into a cell after quite a lot of study. But again, what this does, it allows the viral envelope to fuse with the cell membrane, and then again, the virus is going to come in as a complete virion, sort of, because the complete virion has an envelope, and the envelope is going to be left outside. So what gets in is the nucleic capsid. This thing doesn't have a tail assembly, but it has its nucleic acid core surrounded by a protein coat or capsid. Kind of an interesting twist on this. Well, again, this is just another view of this mechanism. So again, you see HIV binding to the CD4 receptor here on this one, and then the CCR5 receptor binds, and then the envelope fuses with the cell membrane. This is going to allow the virus to come in. Pretty much, it's almost an intact virion, not quite, because it's missing its envelope. But there was a mutation detected in the CCR5, the gene that encodes the CCR5 receptor, on T4 lymphocytes. It's a 32 base deletion. And with that deletion, people that have that deletion, their CCR5 receptor doesn't work. Consequently, HIV can't get in. The CCR5 in a functional state is required for HIV to enter. And so there are people that are resistant to HIV because of the mutation. And this was kind of, it was recognized, and then there was a patient that had acute myeloid leukemia. The patient also had infection with HIV-1. They were treated with stem cells, and those cells were homozygous for the CCR5 mutant. So they could not produce a functional CCR5 receptor. Consequently, after a period of time, the viruses couldn't get into the CD4 cells anymore. They couldn't get into the T lymphocytes, and the person went into remission for HIV. They were without viral load for 20 months. Unfortunately, what killed them was the leukemia. So they were essentially cured of HIV infection or AIDS. Well, anyway, so HIV gets in as an almost intact nucleocapsid. Doesn't have its envelope on, but it still has the nucleic acid core surrounded by a protein coat. And this is typical of animal viruses. They get in as a complete virion. They've got their outside protein coat. So before the viral nucleic acid can become activated, there's another step that has to occur within animal cells. And this step is called uncoating. This is going to involve proteinases that are enzymes within the host cell. The host cell proteinases have to get rid of the protein coat in order for the nucleic acid to be released. So this is a little bit different than what happens with bacteriophage. Now, HIV is a very significant human virus, and it's unique in that it is a retrovirus. When this was first discovered, this was kind of a shocking thing to biologists. And again, it had to do with information, the information pathway. It was assumed that genetic information was encoded by DNA, that DNA was copied to form RNA through transcription, and then messenger RNA was translated to form amino acid chains. But biologists basically assumed you could not take information that was encoded in RNA and use it to make DNA. And along come the retroviruses, and they showed that this was not true. So a retrovirus is a type of virus where the genome is RNA, and they carry within them a viral enzyme called reverse transcriptase. Again, prior to the discovery of this virus, people didn't really recognize the significance of RNA. Since the discovery of retroviruses, biologists recognized that probably the first nucleic acid that existed was RNA, and that RNA was reverse transcribed to get DNA, and then the DNA was copied to make a double-stranded molecule. And it turns out that HIV, the enzymes in HIV, accomplish all of those things. So it is a retrovirus. Again, it has an RNA-type genome. For a while, this was referred to as a viral genome that was double-stranded RNA. It turned out it was not double-stranded RNA. It was just two pieces of single-stranded RNA. So it comes in as two strands of positive sense RNA. Remember, positive sense RNA could be used as messenger RNA. It has the same sequence as messenger RNA, but that's not what happens. Instead, this virus gets in, uncoating happens, and the viral nucleic acid is released into the cell. And then it's going to be acted upon by a viral enzyme that is an RNA-dependent DNA polymerase. Again, the typical name for it is reverse transcriptase. But reverse transcriptase is an RNA-dependent DNA polymerase. So it's going to copy the information stored in RNA and make it into DNA. Again, this comes into the cell inside the viral capsid. This thing, as it goes along, degrades the RNA. So it's not just reading it and copying it, it's taking it apart. It takes apart the RNA, and it uses the nucleotide sequence of the RNA to build DNA, and then it copies that DNA, basically replicates it, to make a double-stranded DNA. So it has a DNA-dependent DNA polymerase function as well. Again, what does a DNA-dependent DNA polymerase do? Replication. It makes the DNA single strand into a DNA double strand. There's a second enzyme that comes, actually there's three enzymes that come into the host, but I want you to know two of them. The second enzyme that I want you to be familiar with now is going to take that double-stranded DNA molecule and it's going to insert it into the host chromosome. This second enzyme is called integrase. This is thought to have potential for gene therapy, because if you can use integrase enzymes, you can take DNA, any double-stranded DNA, and potentially insert it into host cell chromosomes. I'm not sure exactly how the location is determined by integrase, however, because my understanding is the HIV genome can only get in at a specific site. That is the DNA version of the HIV genome, because within a virion, HIV, the genome is 2 strands of RNA, 2 single strands of RNA. Once it gets to this point, it's double-stranded DNA. But here you can see it being inserted into the host chromosome. So the integrase enzyme recognizes a site on the host chromosome, opens it up, and puts the DNA in. Now, once HIV is in the genome, it doesn't come out. Now, this asks, what is a virus called when it's incorporated into the host chromosome? Well, the only viruses you're familiar with at this point are bacteriophage. If a bacteriophage genome is incorporated into the host chromosome, we call it a what? A prophage. Would we call HIV a prophage? No. No, because it's not a phage. So the term bacteriophage applies specifically to viruses that infect bacteria. When HIV goes into the chromosome, it's inserted in there, it is now called a provirus. So it's not a prophage, it's a provirus. But it is going to be copied along with the host cell DNA. If that cell reproduces, it replicates its chromosomes, it's going to replicate HIV right along with it. That is the DNA version of HIV, and it never comes out. So this isn't something that at some other point can remove itself from the host cell chromosome, but then again, the original genome of HIV wasn't DNA anyway. So the DNA, once it's in the host cell chromosome, it stays there. And it's reproduced along with the cell as long as that cell is alive. Eventually, that DNA can be transcribed to encode RNA. So this virus, in order to get into the host cell chromosome, had to go where? Had to get into the host cell, but then it had to migrate into the nucleus, because in the nucleus is where it can bind to the host cell chromosome. But now if the viral DNA, the DNA version of this virus genome is transcribed, then where does the messenger RNA have to go in order for translation to take place? It has to go back to the ribosome. So it has to be able to get into the nucleus, and then it has to be able to get out of the nucleus, and then go to the ribosome for translation. So then we can think about what kinds of viral proteins have to be made. That RNA genome, what was an RNA genome, which is now double-stranded DNA, this is going to encode multiple different proteins. Some of those proteins are the proteins that are going to end up on the envelope. So the glycoproteins, GP120, GP41. There's an enzyme called protease, which you guys don't need to know. And then there are the 2 enzymes that you do need to know, the enzyme reverse transcriptase and the enzyme integrase. The protease that's mentioned there is involved in chopping some proteins into pieces. Never mind, we're not going to worry about those. So the protein components, so first of all, what I said was the DNA was transcribed to encode messenger RNA, which codes for a lot of these different proteins. The other thing that happens is eventually the entire DNA genome is copied into RNA, because the RNA is what has to go into the viral capsid when the virus is eventually packaged up. But after we've got the proteins and then we've made new genomic RNA, what happens next? The step would be the same as the step that occurs in the case of bacteriophage. Once you've got all the protein components and you have replicated the nucleic acid many times over, what happens?
Get out.
Oh, you can't get out yet. You've got parts scattered all over the place in the cytoplasm of the host cell. What has to happen? Remember chaperonins? Assembly. You have to have assembly. So viruses must assemble, and this is an important term because it's what happens, and it's also a stage in the cytolytic virus life cycle. Well, it's a stage in HIV's life cycle too. These things have to be assembled. But HIV is not completely assembled inside the host cell. So the assembly kind of starts, and then the viral parts, they've kind of come to the surface. So what we see in this illustration, the viral nucleic acid is coming along here. We've got some proteins that are going to be making the capsid, but we've got these proteins that have come and bound to the cell membrane. These are the glycoproteins that are going to be the receptors on the surface of the viral envelope. This thing is beginning to get out, and the process that occurs now is called budding, where the virus is pushing against the inside of the membrane pooches outward, and eventually that thing's going to break through. This budding process, here you can see electron photomicrographs, you see a bunch of viruses coming out. This actually makes holes in the cell membrane. So if you make enough holes in the cell membrane, it will kill the host cell, and that's one of the real problems with HIV. Their target is T4 lymphocytes, and they kill them. When we get to the immune system function, we're going to find out about the significance of T4 lymphocytes. They used to be called, well, they're mostly just called helper cells, but they were called helper inducer T cells. Now they're just helper Ts, and their job is basically to make sure that the immune system functions properly. Without T4 lymphocytes, you lose your immune system, not just adaptive or acquired immunity, or excuse me, not just humoral immunity, but cell-mediated immunity as well. Anyway, so these guys get out, they take again a piece of the cell membrane with it. That piece of membrane has the glycoproteins, which are going to be the receptors on the viral envelope. But after the budding process, the proteins that are inside of that thing are going to come together to make the capsid. So that actually happens after budding takes place. And here in this illustration, you can see the viral nucleic acid coming along. You've got these proteins associated. They're in a kind of an interesting configuration here, but it's not complete. After this thing comes out, then this kind of constricts, and now we have our nucleic acid surrounded by a more properly arranged capsid. So that happens after budding. This virus continues to undergo some changes after it leaves the host. And then once they're out, they're ready to start over again, and the process just repeats. So this is a film that summarizes HIV's life cycle. I'll start it up and see where we are with sound.
So this is HIV. It's a typical retrovirus, meaning that it has an outer envelope, and in the center it has 2 copies of RNA, as well as an enzyme here in blue that's reverse transcriptase, which will ultimately turn that RNA into DNA. The virus itself, with this outer envelope protein, actually directly infects T helper cells. The way that it does this is that as it comes up to the cell surface, it uses receptors that are on T helper cells and exclusive to T helper cells, which are CD4 molecule, which really defines T helper cells. It's a surface receptor that binds to the envelope protein. That causes a conformational change and allows a second receptor to grab hold of the envelope. This is the chemokine co-receptor. It's also called CCR5, and we'll talk about that more. What happens now is that the stalk of the envelope protein pierces through from the virus into the host cell and starts to draw the cell membrane and the viral membrane together. And what ultimately happens is fusion of those 2 membranes, and the viral genetic material is injected essentially into the cell, and the envelope protein is left at the cell surface.
Pretty slick, no?
The virus has a matrix and a capsid protein, shown here in green and red, that essentially are digested when it enters into the cell.
That's the uncoded.
That releases the viral enzymes and the viral RNA. And here we have reverse transcriptase, which takes the viral RNA and, using host nucleotides, converts that viral RNA into a single strand of DNA. While it does that, it makes some random errors, which is characteristic of reverse transcriptase. It has very poor proofreading activity. That single-stranded DNA now is again reverse transcribed into a double-stranded DNA. At that point, another enzyme that has come in with the virus in the beginning, called integrase, essentially grabs hold of that double-stranded DNA and carries it through a nuclear pore into the nucleus of the cell. Within the nucleus of the cell, it finds the host chromosome, and it basically, the integrase enzyme makes a nick in the host DNA and allows for HIV to insert itself into the host chromosome. And that right there is what establishes lifelong infection. Now, RNA polymerase comes along and makes messenger RNA. Those messenger RNAs encode for different viral proteins. They end up associating with ribosomes at the surface of the rough endoplasmic reticulum. And here's a piece of mRNA that's making envelope protein, which is directly produced into the endoplasmic reticulum, and it's shuttled then through the endoplasmic reticulum and taken to the cell surface, where at the cell surface it becomes embedded in the cellular membrane, and at this point, coalescing with other envelope proteins that have been produced, you have this cluster of envelope proteins now on the surface of this infected cell. At the same time, there are other messenger RNAs that are being produced that allow for translation of other viral proteins. So here are additional viral proteins being made, which are going to be used to make up the key components that the virus ultimately is going to need. These are transported again to the cell surface, to the area where these envelope proteins are, and a strand of RNA as well as some of the enzymes are part of that complex. This then buds off at the cell surface at this point, but it's still not a mature virion because the polyprotein chain needs to still be digested into its component parts. That's done by an enzyme called protease. Protease breaks up those polyprotein chains and ultimately allows for them to coalesce and form the mature structures that make up the final virion. And now you have a mature infectious virion that can go on now to infect other cells. Once that happens, now the cell can produce tons of viruses, and this is really what then keeps the whole process going.
But that cell, because every one of those virus particles that gets out takes a piece of membrane with it, ultimately that cell's going to be destroyed. So you can't make too many holes in a eukaryotic cell before it stops functioning. And again, you need to know reverse transcriptase, which is the RNA-dependent DNA polymerase, and then also a DNA polymerase that makes the DNA double-stranded. And you need to know integrase, but don't worry about the protease. So the next animal virus that I want to talk about in some detail is the influenza virus. And in association with influenza viruses, we're going to talk about antigenic drift and antigenic shift. So influenza is a lower respiratory tract infection, and it involves this virus or set of viruses. They're in the genus Orthomyxovirus. Most of the genus names for viruses that I talk about here, you don't need to know. So don't worry about virus genus names. The other thing about animal virus infection, when we get to the point where we're looking at a whole bunch of viruses, this will be covered in a matching type situation where you match a specific type of virus with a specific disease type. But before we get to that, we want to talk about antigenic drift and antigenic shift. So what's interesting about influenza viruses is they're not terribly host specific. I was talking about bacteriophage. I said bacteriophage tend to be very host specific. The colophage, the viruses that infect Escherichia coli, are so specific that you can have a specific bacteriophage that will only infect a particular strain of E. coli and not another strain of E. coli. This is why phage typing works. Remember phage typing? Oh boy, that was way back in the early part of the semester where I said if you had known types of virus, you could use those known types of virus to identify types of bacteria. It only works because bacteriophage are very host specific. But apparently animal viruses are much less host specific. And this is true because if you think about rabies, rabies is a viral disease, but the rabies virus, the babesioviruses that cause rabies, can infect a very wide range of animals. They're not nearly as host specific. As soon as I say that, and I think, actually, the reason for that is because all eukaryotic cells are amazingly similar to one another. And so when I say there's all these different types of animals, we are just not that different genetically from other animal types. But anyway, the reservoirs for the influenza viruses are humans, also swine, and different types of birds. So you see some host organisms there. This particular virion, the influenza A, is an enveloped virus. So again, you're going to have nucleic acid surrounded by a protein coat, and outside the protein coat is envelope. That envelope was cell membrane taken from the last host that virus was infecting. They tend to get out via budding, just like HIV did. But the genome for influenza viruses is more complex. So HIV, the genome was 2 pieces of RNA, 2 single-stranded RNA molecules. The genome of the influenza virus is 8 single-stranded RNA molecules. So there's more segments. These influenza viruses are categorized based on the proteins that move to the surface of the envelope. So they're proteins in that version of HIV infection we saw on the film. The viral genome encodes proteins which move to the host cell surface, and those will ultimately end up on the envelope of the virus. Well, the same thing happens with influenza. There's 2 different types of proteins that are used to identify the different types of influenza virus. One of the proteins is called hemagglutinin. We're going to talk about hemagglutination later in the laboratory. Hemagglutination is what causes blood cells to clump. So hemagglutinin is a protein that will clump red blood cells. The other protein is called neuraminidase. And neuraminidase is an enzyme that breaks the glycosidic linkages of neuraminic acid. You don't really need to know that. All you need to know is that these proteins are on the surface of the viral envelope. So you have hemagglutinin proteins and you have neuraminidase proteins that are sitting on the surface. They're going to serve as receptors. They're going to allow that virus to bind to the next host. But there's a lot of variation here. It turns out that these influenza viruses then are identified through serological typing. Again, back to stuff you learned early in the semester. Serological typing means you use antibodies to detect specific antigens associated with infectious agents. The antibodies are proteins that we produce. We'll learn more about these when we get to the section on immunology. But we produce antibodies, and antibodies tend to be very specific in their interaction with antigens. So the antigens, in this case, are these hemagglutinin proteins and neuraminidase proteins that are on the envelope of the influenza virus. So these things are identified. Turns out there's 16 recognized types of hemagglutinin and 9 different types of neuraminidase. And these can combine in different arrangements on the surface of the envelope so that you have many different strains of influenza virus. And these things are referred to as subtypes or serotypes. Serotype is just short for serological type. Serology is the study of antibody and antigen interactions in vitro. I think I gave you that term when talking about serological typing. So serotype is, again, just short for serological type. But these things have the letters H for hemagglutinin, N for neuraminidase, and then they have numbers assigned based on which segment of that particular protein type you're looking at. So you may have heard of an influenza virus that was identified as the H1N1 strain. That means it had number 1 type hemagglutinin and number 1 type neuraminidase. You get different combinations of these things that make the different influenza viruses different. Well, in order for the body to respond to an influenza virus appropriately, that is, make antibodies against that particular influenza virus, it has to interact with that virus in some way. If you're lucky, you get the flu vaccine, and the flu vaccine introduces the virus to you, but in a form that can't infect you. And typically now, when influenza virus is administered, they try to protect you against more than one strain. So the CDC pays attention through the World Health Organization. They try to keep track of which viruses are developing. Typically they develop someplace in Asia because you have people and birds and pigs living in close proximity. At any rate, they try to keep track of which versions of the proteins you need to be protected against. But this is tough. It's really tough because of antigenic drift and antigenic shift. Antigenic drift, again, the antigen is that protein on the surface of the envelope, either hemagglutinin or neuraminidase. Proteins are made up of amino acids. Amino acids are encoded by nucleotide sequences, codons, right? You have different codons that code different amino acids. Well, antigenic drift refers to mutations in the nucleic acid that can lead to a change in the amino acid sequence. Mutations happen. Mutations are a characteristic of life, and even though viruses are not considered living by most references, they're right on the border. We've already said viruses have many of the characteristics of life, and one of them is mutation. Viruses mutate. With HIV, they mentioned the fact that reverse transcriptase doesn't have very good fidelity. When it copies the information from RNA into DNA, it makes mistakes. And so there's going to be changes in the nucleotide sequence. The same thing can happen when you're making the genome for the influenza virus. So antigenic drift is going to be a change in the envelope protein, but that change is due to a mutation of the genome. Those mutations happen. They typically cause relatively minor changes in the surface protein, but those minor changes might influence how well the virus can bind to a host cell, or which type of cell they're likely to bind with. This is probably why you have more than one host for influenza viruses in the first place. So, again, as I mentioned, it's just like the HIV in that the viruses are encoded by the viral genome, the virus proteins, hemagglutinin and neuraminidase. They're encoded by the viral genome. They move to the surface of the host cell as antigens or protein groups on the surface, and they're going to end up on the envelope when that virus goes out by budding. Mutations are going to change the exact composition of those proteins. You change the amino acid sequence. Now, these two proteins, hemagglutinin and neuraminidase, have specific functions, but they are not like the enzymes that are required to run metabolic processes. Typically, if you change those enzymes that are doing some specific catalytic activity, you change them very much, they don't work. These things can be changed quite a bit, and it really doesn't matter, because all they are is receptor sites on the surface of the host. But they may influence how well that virus can bind and how well it gets in. There's always selective pressure, so there's lots of different viruses being produced. Immune responses allow you to produce antibodies against certain ones of them. If you make antibodies against certain types, you're likely to eliminate them from the population, which then puts pressure on the selective pressure. The ones that survive are the ones that you can't produce antibodies against, or for some reason you don't. Anyway, so antigenic drift is change in the antigen due to mutations of the viral genes. Antigenic shift is a faster moving process because antigenic shift involves reassortment of viral genes. Again, there were 8 segments of RNA that get incorporated into the virion. Those 8 are not necessarily coming from the original or the same virus. So what happens is within a host, if you have viruses that are possibly from alternate hosts that have gotten in there, you can take segments, put together different combinations of RNA, and you end up with quite a big change in those envelope proteins. So this is showing what can happen where you have a human strain of HIV and a highly pathogenic avian strain, and what's happening is these highly pathogenic proteins from the avian strain get mixed with the human strain, and you end up with a virus that is a highly pathogenic human strain. And then influenzas pass from birds to humans. I believe that the 1918 influenza epidemic, which killed a very large number of people, originated as influenza swine. So it was a pig disease, and then it was passed to humans. probably due to antigenic shift. So both antigenic drift and antigenic shift bring about change. Drift is slower because it involves mutation. Shift involves this reorganization or reassortment of viral RNA segments where you put together multiple within the same virion. Either way, it's going to make it tough for the body to produce antibodies against these viruses and protect you from them. This is showing mutation where you have a little bit of change. Here you see a virus that has these purple receptors on the surface or coat proteins, and they get changed a little bit, but they're still purple. Over here, we put purple, green, and yellow together, and we end up with a virus that has a completely new characteristic because it's a combination of multiple different RNAs from different strains. So what's on the left is antigenic drift, what's on the right is antigenic shift. And you need to know the difference. Okay, now what we're going to get into is specific types of viruses that can infect humans and cause disease. In microbiology textbooks, these are usually organized by region of the body. In fact, microbiology textbooks tend to put all kinds of pathogens into regions. Again, this has to do with what type of tissue, what type of cell can a virus infect. What do you think makes the difference? Why can some viruses infect blood cells? Some viruses infect nerve cells. Some viruses cause gastrointestinal infection.
Yeah, maybe I guess. Is it because they have various different types of receptors?
Exactly. So when we're talking about HIV, you have a specific receptor, glycoprotein 120 and 41, that have to bind with specific receptors on the CD4 lymphocyte or T4 lymphocyte. Different viruses have different receptors, so it's during that adsorption phase or attachment phase when the virus is making contact with the host. The receptors have to bind to the host cell surface, and that's going to make viruses capable of infecting only certain types of tissue. So they are not only host specific, they're tissue specific. So we're going to start out with viruses that tend to cause skin infections, but usually associated with skin, we also put in oral and genital regions because your skin kind of folds into your mouth, folds in other regions. Well, anyway. So the first one on our list are the human herpes viruses. And the family here is Herpes viridae. In terms of genus name, these are called herpes simplex virus 1 and herpes simplex virus 2, or HSV1, HSV2. These typically cause both oral and genital herpes. HSV1 usually causes oral herpes, and HSV2 typically causes genital herpes, but they can cross-infect. So it's possible to get a virus that is normally an oral virus on your genitalia and vice versa. Here we see the effect of oral herpes. Probably most people would recognize these lesions. People say, oh, you've got herpes. Or you might say, you've got cold sores. I believe the connection had to do with if you get a cold, if you catch a cold, sometimes the viruses that cause the common cold will activate human herpes viruses. But these viruses, human herpes viruses in general, are viruses that go into a latent state. So if you're infected by these viruses, you keep them. They tend to become associated with neurons, and they stay present in the body in a latent state, an inactive state, throughout your lifetime. So these things can reactivate. These appear to be the mouths of relatively young individuals, which is sad. They probably got that virus from their parent. It's pretty easy to accidentally transfer these viruses by using the same cup or spoon, that kind of thing. Or parents kissing their offspring on the mouth. So these things infect sensory neurons, and again, they remain throughout the life of the infected individual, but in a latent state. So they are a type of herpes infection. It's a type of infection that's considered latent. The virus is inactive, typically for very long periods of time, and then they can reactivate at various points. And again, I think the reason that the lesions on the mouth are called cold sores, when people would get a cold, something about being infected by the rhinoviruses would activate the herpes viruses. Here we see genital herpes lesions. So the lesions on the genitalia don't look as severe. but these are relatively painful. And typically infection is going to occur through direct contact, but it can be indirect if it's a short period of time between exposure from one individual to the next. Here we see herpes virus infecting, again, young individuals in regions of the body where they're not normally expected to be found. So we have eye infection, infection associated with the nose, right along the edge of the nasal cavity, and on the fingertips. So these viruses don't necessarily infect only specific regions of the body. And they cause very painful lesions. If you've ever had an active cold sore, they're very, very uncomfortable. They're doing a lot of damage to the tissue. Now, another human herpes virus type is human herpes virus 3. Human herpes virus 3 is also called the varicella zoster virus. It's more commonly referred to as the varicella zoster virus. Varicella is chickenpox and zoster is shingles. So this is the virus that causes both chickenpox and shingles. And again, it's a human herpes virus, so once you've been infected with it, you keep that virus throughout your life. Chickenpox, varicella, is a relatively mild disease, and so there was a period of time where parents, they kind of kept track of the neighborhood, and when somebody got chickenpox, if they had a child that hadn't had chickenpox yet, they would get them together with the neighbors and everybody would go visit so that they could get chickenpox. Because it was thought that if you got chickenpox, you only had it once and then you couldn't get it again. What they didn't know is that when you got chickenpox, you had the virus and you had it for life. Well, it could either reactivate later as zoster, shingles, or it could reactivate later as chickenpox. And I had a student one semester that told me she had had chickenpox 3 or 4 times. Her physician said, that's not possible, you can only get chickenpox once. And she said, okay, the next time I get chickenpox, I'll come see you, which she did. Her herpes virus 3, human herpes virus 3, was reactivating as chickenpox, and it did this over and over again. Why? I don't know. But again, there's a lot of variations on a theme. So shingles, zoster, this typically occurs in older individuals. However, it was showing up in young individuals, and zoster is not pleasant at all, because what you get are lesions very similar to the lesions of cold sores, where you have this blistering-like effect and then a certain amount of fluid drainage. It's very, very uncomfortable. Typically occurs along nerve trunks, and it can take place across fairly wide regions of the body. So this is very painful. In association with these lesions, there's typically high fever, and the person just they feel really drained. They're not feeling good at all with shingles. Well, because shingles was showing up in young individuals, it was determined that rather than allow children to continue to go visit their neighbors and get chickenpox, that it was time to come up with a chickenpox vaccine. So now there is a chickenpox vaccine, and that's a much better way to control that virus. And there's more than one anti-shingles vaccine for elderly individuals as well, to prevent that virus from reactivating. There are human herpes viruses 4, 5, 6, 7. Some of those are associated with, well, I'm trying to think, what are they associated with? The viruses that are associated with mononucleosis fall into that category. And then there's another, well, anyway, they're not as specific in terms of number going with a specific infection, but human herpes virus 8 causes Kaposi sarcoma. This is a tumor on the skin surface that typically showed up in people that were infected with HIV. So this was kind of a characteristic feature. If people had HIV infection, they would oftentimes, the first symptom they would get would be Kaposi sarcoma. Okay, so those are the human herpes viruses. You need to know 1, 2, 3, and 8. Never mind the ones in the middle. Next, we're moving on to the paramyxoviruses. Again, the family is Paramyxoviridae. You don't need to know those either. In this category, we have measles viruses. Measles, the genus is morbillivirus. Again, you don't need to know the genus names of these viruses. What I'd like you to recognize is that the measles virus, the other name that they go by is rubeola. I think because these terms have been in the clinical setting for a very long period of time. So measles viruses are referred to as rubeola viruses. Measles is an acute infection characterized by a rash, usually fever, sometimes respiratory symptoms. The rash is very itchy and uncomfortable. And in some cases, that virus can cause neurological damage. So it can damage the eyes and whatnot. Another virus, somewhat related, primarily, it's not really related, but it's related because of the vaccine that's produced against these viruses. The virus is called the rubella virus, and it causes mumps. Now here we're incorporating mumps with the viruses associated with skin and also mouth. Actually, mumps is associated with the gastrointestinal system because it's an inflammation of salivary glands. And the salivary glands are accessory organs to the GI tract. So this is a little bit out of place. But anyway, these viruses, the rubella virus, cause enlargement of the salivary glands. Here you see individuals, either one side or sometimes both sides would be very swollen. This is uncomfortable in young individuals, more of a problem in older individuals because this virus, again, is not completely restricted to the salivary glands. And so older individuals, if they hadn't had mumps as a child, they can have infection with this virus and it can get into the prostate gland and cause serious difficulty. Consequently, children are typically immunized against both measles and mumps with the MMR vaccine, measles, mumps, and rubella, which is German measles. So German measles, sometimes also called 3-day measles, again, it's an acute disease. It's caused by rubella virus. So the genus is rubevirus, but again, the common name is rubella. So if you heard of somebody having rubella, it's German measles. Don't worry about togavirus or togaviridae. Concentrate on specific aspects of the name. Rubella is, you'll see this as a matching situation with German measles. Again, the symptoms are generally mild with this one, rash, fever, sometimes a sore throat. But this virus, if it gets into a woman, a pregnant woman, it can cross the placenta and cause severe nervous system damage. So it's dangerous to pregnant women. It can cause blindness, it can cause deafness, it can cause damage to the heart, damage to the genitalia, etc. So the German measles virus, rubella, is much more dangerous under certain circumstances. Again, this is why there is now a vaccine to protect against these 3 types of virus, the measles, mumps, and rubella, and it is the MMR vaccine. These are all highly contagious, so non-immunized individuals put other people at risk. Oftentimes, there can be members of the population that for whatever reason, maybe their immune system is not functioning properly, vaccines don't work for them, and so if the rest of the people are not immunized, they put those certain individuals at risk from disease development. We'll talk about herd immunity when we get to the section on epidemiology. The next type of virus we're talking about is the pox virus. So there were a group of viruses called variola viruses that cause smallpox. This virus, well, the disease caused by this virus, smallpox, was actually eradicated. The World Health Organization launched a campaign. They recognized that smallpox was a disease that only infected humans, and so they launched a campaign to immunize everybody against smallpox. And they declared smallpox eradicated in 1979. I know this date simply because my oldest son was born in 1980, and he did not get a smallpox vaccine. Myself, I have a smallpox vaccination scar, and many adult individuals have their smallpox vaccination scar. Unfortunately, there's a kind of a downside to this because the U.S. military personnel are now given smallpox vaccine. And the reason they're given smallpox vaccine is because it's assumed the smallpox virus, although the disease was eradicated in 1979, the virus itself was not. The virus exists in various freezers, I think in the United States and in some other countries, and scientists debated whether that virus, that genome should be eliminated, as in should we knowingly eliminate a naturally existing entity, and it was determined that no, we should not. And so it was not eliminated, and consequently, it is a potential germ warfare agent. And that's why military personnel are immunized against it. Well, okay, moving right along. So the next virus is the papillomavirus. In this case, the genus is Papilloma. That's kind of a term that is put together from various pieces of other terms. This virus, the papillomavirus, does cause warts, but it's more significant because it also causes cancer, cervical cancer and cancer in other parts of the body. Now, we haven't talked about cancer. Cancer is brought about by genes called oncogenes. Oncogenes are genes that can, through modification, lead to tumor formation. And apparently the papillomavirus can cause cells to convert into tumor-producing cells, and this is the real problem with it. So although it does cause genital warts, it also causes cervical cancer. It's very easily transmitted. So according to the CDC, about 14 million people become infected with human papillomavirus every year. It's sexually transmitted. And as a consequence, they decided that they should put together a vaccine, the HPV vaccine. And so it causes cancer of the cervix, but also the vulva, the vagina, the penis, the anus, the mouth, the throat, the oropharynx, in addition to warts. It's the cancer-producing ability of this that's really the problem. The trade name for the vaccine is Gardasil. I'm not sure if that's the only one. And initially, Gardasil was only going to be used for young individuals, so individuals that were prior to when they might become sexually active. The idea would be to immunize them before they became sexually active, and then when they became sexually active, they couldn't get infected. Well, it didn't take too long for older individuals to say, wait a minute, I'm not infected and I'd like to be vaccinated too. And so then they had to expand who could get the vaccine. I believe now it costs you money, but as an older individual, you can still get that HPV vaccine. Okay, so now we're moving to diseases of the gastrointestinal system and related organs, except for mumps because it got incorporated with the skin infections. Notice that each time I'm giving you a name of a type of virus and the family, most of the time you don't need to remember those. They're not something you need to know. Now we're going to be talking about hepatitis viruses. The hepatitis viruses typically are abbreviated H for hepatitis, and then A for whatever letter is designated to them. So this is the hepatitis A virus, but there's also a hepatitis B virus and a hepatitis C and D and E, and the letters go on and on and on. There's a whole bunch of these things. So hepatitis A virus is HAV. The genus is hepatovirus, but again, recognize it as hepatitis virus. These cause inflammation of the liver. If it's hepatitis A, this one is most commonly acquired through contaminated water, possibly contaminated food, and it's usually, although it causes damage to the liver, it's usually not life-threatening. It's kind of a self-limiting disease. Here we see hepatitis A virus, electron photomicrographs, and then a model of the virus, and then a person that shows evidence of jaundice, so the yellowish color of the skin, the yellowish color of the whites of the eyes. The liver's being damaged, but again, typically this can be self-controlled. The hepidNA virus, this is the virus that causes hepatitis B, and this one's much more dangerous. This is the one that's sometimes called serum hepatitis because it's usually transmitted from host to host through contaminated needles. This one can cause severe liver damage and can be potentially fatal. Consequently, there was a vaccine developed for hepatitis B fairly early on to immunize people against this one, recognizing that it was potentially fatal. And then there's hepatitis C virus. What you'll notice if you were paying attention to the family up at the top, this is a completely different family. So there's a group of viruses that are all called hepatitis viruses. They're assigned hepatitis virus letters, A, B, C, D, E. They're not closely related. And many different types of virus, the only thing they have in common is they cause inflammation of the liver, damage to the liver. Hepatitis C, again, causes hepatitis the hepatitis C virus causes hepatitis C. Causes damage to the immune system. For a long time, they weren't sure exactly how this thing was transmitted, but apparently hepatitis C is transmitted by objects that are used between people, things like using common toothbrushes, using common razor blades, okay. And now there's a vaccine against hepatitis C. Now we're moving to diseases of the respiratory system. One that everybody has had experience with is the virus that causes the common cold. This says 95 to 100 different members of the genus rhinovirus. This one, it is useful to know this genus because if you think of a rhinoceros, it causes you maybe to think of your nose. Rhinoceros has a big, point on the nose. So you think, rhinovirus, yes, rhinoceros, something to do with the nose. The common cold, your nose plugs up. This is an upper respiratory tract infection that tends to make your whole face feel like it's swollen up. What you see on the left-hand side, that's a backlit photograph of somebody sneezing, and those are called droplet nuclei, so they're being dispersed into the air. These viruses are very readily transmitted from person to person, and the common cold is much more common during the wintertime. Anybody know why?
Because people have huddled together in the winter?
Yeah, because people get huddled together. That used to be that people said, well, you're going to catch a cold if you go outside in the cold. Don't go out in the cold because you'll catch a cold. Actually, you're probably better off to go out in the cold. Stay away from the people. It's the people that are all clustered together inside. That's where you get the common cold. You get it from somebody else that's already got it. This particular virus is not one that we immunize against, in part because there's so many different strains of it, but also because it's not a disease that kills people. It just makes you very uncomfortable for a period of time. Again, you've all experienced the common cold. Then we come to the orthomyxoviruses. I already talked about this, the influenza virus. Influenza is a lower respiratory tract infection, so much more serious. Sometimes secondary infection can occur in association with influenza, and then people get bacterial infections and develop pneumonia. But again, the orthomyxovirus that causes influenza can infect humans, swine, birds. We talked about antigenic drift and antigenic shift. The genes encoding hemagglutinin and neuraminidase are multiple. You have different versions of them, and as a consequence, you can get changes in the surface antigens either through mutation, which is antigenic drift, or through recombination. When H1N1, this was a couple of years ago that the H1N1 variety of this virus was present within the population, the CDC worked really hard to try to immunize everybody against it. And at the time, at microbiology conferences, people talked about the shortages in this country of companies that can produce vaccines. And they said part of the problem is this vaccine production typically involves the use of eggs. And there's only so many eggs available to make virus that can be used to make a vaccine. You have to get the antigens that you would produce antibodies against. And part of the reason that the CDC pushes so hard to get people to be immunized against HIV up front is that if an epidemic were to occur, there's no way that all the people in this country can be immunized as an emergency response to an epidemic. We don't have enough vaccine. We can't get it fast enough. We can't produce it fast enough to immunize people in the event of an epidemic. And that's why they want you to be immunized ahead of time. Try to get the vaccine out into the population. It takes a while to get everybody immunized. If you haven't done it yet, it's coming up on influenza season. Again, this is a type of virus that's transmitted from person to person when people are in close quarters. So during the wintertime, this is when more people are going to get influenza or potentially get it. If you haven't been immunized, you should be.
Is it the flu shot?
That's your flu shot. So sometimes people talk about having stomach flu. That's not influenza. Influenza is a lower respiratory tract infection. If they have a stomach ailment, it's something else. There's a lot of different viruses that infect the GI tract, but not influenza viruses. Now, there's a group of viruses called coronaviruses. These were identified a fairly short time ago They cause what's called severe acute respiratory syndrome, or SARS. This disease killed close to 800 people during outbreaks in 2002, 2003. And this was a virus that spread fairly rapidly from regions in Asia. Cases occurred throughout the world. And what was impressive about it was how quickly they came up with a method for identifying these viruses using basically DNA analysis. So they now know how to identify this type of virus very quickly when it shows up. Next virus type are the Bunya viruses. Now, again, ignore the Bunya virus and the Bunyaviridae. The name I want you to pay attention to here is the genus name Hantavirus. There are quite a number of viruses that were named according to rivers in whatever country they were first found in. So Hantavirus is named for the Hantan River Valley, or the Hantan River. This is a virus that causes pulmonary syndrome. So it's Hantavirus can cause a potentially fatal respiratory disease fairly quickly. This virus is associated with rodents and apparently transmitted in fecal material and urine that is deposited by rodents when they're living in close proximity to people. People run into the Hantavirus when they're cleaning up. Now, they may be cleaning up a building that has had rodents inhabiting it, or they might be camping in a situation where rodents have Hantavirus. But it's potentially deadly, this, because it is a hemorrhagic fever type virus. Now we're moving to viruses of the nervous system. And the first example here is the viruses that cause poliomyelitis, or polio. These are enteroviruses. There are multiple different types of enteroviruses. They cause damage to motor neurons, and typically this will cause permanent paralysis in individuals that are infected with polio. This is a disease that in this country most people are immunized against. But in other parts of the world, lots of people have polio. Even though there's a vaccine available, it's not necessarily available to all people. Fortunately, the vaccine that was developed for polio can be given orally, so it's pretty easy to administer. And oftentimes, children were given just drops, or they were given drops on sugar cubes so that they would take it. The other thing that the World Health Organization is trying to do is come up with a way of producing vaccines that doesn't involve eggs. So they'd like to be able to use tobacco plants, eukaryotic organisms that can be grown in abundance, and they would be able to grow the viruses up and then make vaccines that way. I don't know how well that's progressing. The next type of virus are the encephalitis viruses. Encephalitis is inflammation of the brain. And there are multiple different types of encephalitis viruses, so they're not necessarily all closely related. There's a term that is applied to some of the viruses that cause encephalitis. The term is arbovirus. And arbovirus just means arthropod-borne. So these are viruses that are transmitted by arthropods, typically mosquitoes. So they're vector transmitted. But again, there are multiple different types of encephalitis viruses. So which one do I want you to know? Not that one. I'll come back to that. I want you to know about flaviviruses. This is a virus that I want you to know the genus name. So the genus name is rhabdovirus. The rhabdoviruses are transmitted typically by a bite of an infected animal, and they cause rabies. Now there's a bit of a fallacy here because most people think you have to be bitten by a rabid animal in order to contract rabies, and that's not true because animals lick their fur surfaces. If you handle an animal that has been licking its fur and it has rabies, you can pick up the rabies virus that way. This is a virus that most domestic animals, if they're spending time outdoors, they're supposed to be immunized against the rabies virus because in order to detect rabies virus, if an animal hasn't been immunized and it bites somebody, the means for detecting it involves disposing of the animal, taking the brain tissue, and then the brain tissue is sliced into very small amounts and put on slides, and they use fluorescent antibodies. The fluorescent antibodies can be viewed with a fluorescent microscope, And what you see here is fluorescent antibodies binding to rhabdoviruses that are in this tissue. But it requires the animal to be sacrificed. So if you have an animal you care about and it spends time outside, get it immunized. You don't want it to bite somebody and then find out, oh, by the way, you have to kill that animal now. Because that's how we determine has it potentially got rabies or not. And some of the veterinary clinics, I think, offer rabies virus free. Well, actually, I know this also because our neighbor, for some reason, people come by our place and they deposit cats. So early in the spring, there were 5 cats deposited. And at some point, the neighbors decided they didn't want these cats to reproduce, so they started trapping the cats and taking them to the SPCA, and they would snip off the end of their ear and immunize them against rabies. But they did it for free. So rabies virus is a vaccine, at least sometimes is free. The ear snipping wasn't so nice, and they neutered them also. But they got all except one female, and of course it got pregnant, and then it had 5 more cats. And I was like, this is crazy. These animals just keep reproducing. Yeah, they do that. Okay, this is the other virus that I want you to know that causes encephalitis. So flavivirus. The flaviviruses, the West Nile fever is caused by a flavivirus. This is primarily a disease that infects animals other than humans. So it's usually a bird disease or a horse disease, but it can be transmitted to humans. So there's our mosquito vector, which is the mosquito that we looked at in the laboratory, Culex pipiens. They're the primary transmitters of flaviviruses. And again, flaviviruses cause encephalitis. Then we come to diseases of the circulatory system. Again, a flavivirus can cause yellow fever or dengue fever. So these viruses in this group, the flavivirus group, the genus Flavivirus, can cause more than just encephalitis. Dengue fever, yellow fever, typically headache, fever, muscle pain, bone pain, but sometimes these are hemorrhagic viruses, and so they can cause internal hemorrhaging, which is potentially lethal. Again, their vectors are mosquitoes, not Culex. The mosquito that is the vector for the yellow fever virus and the dengue fever virus is in the, I think it's Aedes aegypti is the vector. Anyway, dengue fever, now there's a form of this called dengue hemorrhagic fever, again where it causes internal hemorrhaging, which can lead to circulatory shock. It's potentially fatal. This is a type of viral infection that people are very concerned about due to global climate change. Because as climate change is occurring, regions where this virus didn't exist, now the virus doesn't exist. So it's expanding its range. The mosquitoes are also expanding their range. And this is a very dangerous type of virus. Then we have Zika viruses. This is again caused by a flavivirus. This was believed to be originally a disease of monkeys, but it became a disease of humans very recently, 2015, 2016. Again, here we see the name of the mosquito that is the vector, Aedes aegypti. But this virus can also be transmitted sexually and through blood transfusion. So the Zika virus, this is a positive strand RNA type virus that can be translated into proteins immediately. So a positive strand has the same nucleotide sequence as messenger RNA, gets into the host cell, it just goes to the ribosome and gets translated right away, encoding proteins. Most people that are infected with Zika don't have any symptoms at all. Sometimes people may get a little bit of fever, headache, joint pain. They might not know they've been infected. But again, the real damage is if infection occurs in association with pregnant women, because then it can cross the placenta and cause damage to a fetus. And it can result in microcephaly, which is usually deadly within a short period of time. So it causes severe damage to the brain. This is a virus that now is in the United States. So I'm not sure how many states, Florida, I'm not sure which ones. But anyway, CDC recommends that people that have traveled to areas where this virus exists should be screened to see if they've been exposed to it. And apparently in some parts of the country, they're just encouraging people not to get pregnant. Not this country so much, but in other countries, don't get pregnant because of Zika. I doubt if it'll actually impact the human population, however. This is talking about how much of the virus is present in the bloodstream. In serum, it's only present for 1 to 3 days, in saliva 3 to 5 days, in urine up to 11 days. After that, you're not going to be able to detect it. And so depending on when you get screened relative to when you travel, you might not recognize it anyway.
So does it stay in your body for a long time, like some of these other ones that are, in your body forever? Like,
I don't know how long it stays, but apparently it's not detectable. So does it, disappear into nervous tissue? I don't know. This virus, the filoviruses, family Filoviridae, cause Ebola. So a filovirus, this one I do want you to know the genus. The filovirus is a kind of a string-like virus, so think of a filament. They don't have a typical viral shape when we think of other types of virus, but they're these long skinny strings. And Ebola is hemorrhagic fever. This was information about Ebola in Africa. This was a very severe type of an epidemic and very difficult to control because of the region where it occurred. They really didn't have measures to prevent it. I believe what happened is the CDC worked really hard to come up with an Ebola vaccine very quickly. And usually vaccine development requires a long period of time because they have to develop a prototype vaccine and then test it for a number of years. In this case, they didn't want to take the time to test it for a number of years. They wanted to get it into the field immediately. So there's information on the ASM site and there's information on the CDC site about Ebola. And then we come to the retroviruses. The last one, the human immunodeficiency virus, or HIV. Again, the genus is retrovirus. They're the only retroviruses you need to know. And they cause acquired immune deficiency syndrome, or AIDS. They do this by wiping out the T4 lymphocytes. Without T4 lymphocytes, your immune system fails. And people typically die from infection with something else. But it's because the virus has destroyed their immune system. This last little bit is about viruses and cancer. So cancer is a disease commonly associated with aging individuals, or I should say it used to be commonly associated with aging individuals. Again, real concern is being shown now because cancer is showing up in young individuals. And part of the reason for this, I believe, is because we're exposed to carcinogens, mutagens, chemicals, not one at a time, which is how they're tested, but lots of them all at once. Our environment is full of potentially cancer-causing agents. We have within us segments of DNA called oncogenes. Oncogenes are genes that can convert normal cells into tumor cells. And if they become tumor cells, then they have some characteristics that are not like normal cells. But for every oncogene, you have 2 oncogene suppressors. The job of the suppressors is to produce substances that prevent the activation of the oncogenes. So you have this system set up. Unfortunately, if you lose one or more of your oncogene suppressors, then the oncogenes will produce tumors. So the oncogenes can be cellular in origin, they can be viral in origin. If they're cellular, they can occur due to mutation of the cells. The viruses can carry DNA in and change them and make them into oncogenes. And so there are a number of viruses now that are recognized as being oncogenic. That is, if you're infected with these viruses, they're likely to activate your oncogenes and cause them to form tumors. And tumor cells behave differently. So if they're altered, then, can I finish this? Or do you want to?
You mean the sentence?
Yeah, we're almost done. I'm trying to finish this and we're almost done with lecture and then they want to run away.
That's fine.
All right, okay, hang on there.
So
So,
how long is almost done? 3 hours?
no, we're supposed to be finished at
I know how she works.
10 to 7, so I just want to get through these last
Hey,
slides.
better keep moving.
Okay, so, so if cells are altered so that they become tumor type cells, they have some abnormal characteristics. For one thing, they become immortal. They don't die. Typically, eukaryotic cells, human cells, have a lifespan and then they conk out. But these things just keep growing over and over and over again. They decrease their dependence upon anchorage. So a lot of times, cells that are associated with specific tissues tend to stay put. They stay associated with that specific tissue. The cancer cells, the tumor cells, don't. So they can metastasize. They can get into the bloodstream and they travel all over the place. The other thing is they lose their contact inhibition of growth. So when they get to a new area, rather than fitting into a tissue and being restricted in terms of how much they can reproduce, they just reproduce like crazy. Once they get established in an area, then they tend to take over the circulatory system so that they tend to induce the development of blood vessels to bring them more nutrient, to bring them more oxygen so they can continue to grow. And you get these masses of tumors with these immortal cells that just keep growing and taking resources and growing and taking resources. And to me, cancer is very much like the human population on planet Earth. Scary. Growing and taking up resources in an uncontrolled way. It's scary stuff. Anyway, so here are some viruses that are known to be associated with tumor development. The human herpes viruses, Kaposi sarcoma. Human papillomaviruses, the viruses that cause cervical cancer. The hepadNA viruses, not only do they cause hepatitis, they cause liver cancer. Herpes viruses that are the numbers between 3 and 8. So I said you need to know 1, 2, and 3, and 8. But there are human herpes virus numbers in between that cause Burkitt's lymphoma, nasopharyngeal carcinoma, Hodgkin's lymphoma, etc. You don't need to know their numbers. And then the retroviruses that cause HIV can also cause leukemia, and they can cause tumors in other parts of the body. This business about the unpasteurized milk, we don't really know what causes breast cancer, but in mice, there's a type of tumor, a breast tumor, that is transmitted through milk. And so researchers thought, wow, what if human breast cancer is caused by a virus that's transmitted in milk? And they hypothesized that it might be a virus associated with cattle because we consume mostly cow milk. And then they thought this virus would be much more likely to occur in people that consumed unpasteurized milk than people that consumed pasteurized milk. I don't know if they really made a connection between, it was the bovine leukemia virus that they wanted to suspect of being the cause of human breast cancer. I don't know what happened to that study. We've already talked about viroids and prions. Never mind. Okay. Again, this material is not on exam 2. It's on the final. And we're still behind in lecture, huh?
Yeah, I don't think so.
No?
Cars are always.
More.